Hello, good morning, everyone. Welcome. We are going to start the technical sessions of materials and manufacturing. Well, as, as our first speaker, we have, the authors are Gabriela Rojas, Abel Zapa, Daniel Loa, Andrea Rivas Castillo, Abril Ángeles, Luis Ángel Tejeda, and the title of the work is Assessment of Cadmium Sulfide, I'm sorry, Assessment of Cadmium Sulfide Nanoparticle Synthesis by Cadmium Tolerant Fungi. Well, I hope he helped us to tell us the title correct. Okay, I think Daniel is going to present. Daniel, are you ready?
Yes, I'm ready.
Okay, well, the stage is yours.
Thank you. Thank you very much for the presentation. And well, our project is entitled Assessment of Cadmium Sulfide Nanoparticle Synthesis by Cadmium Tolerant Fungi. And as introduction, we have that semiconductor nanoparticles such as cadmium sulfide nanoparticles are a material composed by a metallic and non-metallic element which have a high electrical and optical stability. And this is due that they have a valency and conduction band that are separated by a specific distance known as band gap energy. And this band gap energy increase when the material reduces its size to a nanometric scale, and the electrons that are in conduction band when are excited by photon return to valency band and release energy with high brightness and intensity. And for those reasons, these kind of nanoparticles are different applications such as in solar cells, cutting of electrodes, biological sensors, and fluorescence microscopy. Cadmium sulfide nanoparticles could be biosynthesized by intracellular or extracellular process. Here, ions or metal ions are captured by cells, and sulfur is reduced to combine with ions or with cadmium ions to form cadmium sulfide nanoparticles. And this process is carried out by cellular stress with the aim to reduce the metal toxicity or reduce the metal solubility. And the process that involve these mechanisms are absorption, secretion, conjugation, and precipitation. And some responses involved are active transporters, membrane proteins, cysteine-rich molecules, reductase enzymes, and metallothioneins. And for previous reasons, cadmium-tolerant fungi are promising organisms because they are adapted to adverse conditions to avoid or reduce metal toxicity or reduce the metal solubility. And for previous reasons, cadmium tolerant fungi are interested in this topic because they're involved in cadmium sulfide nanoparticles, unlike another fungi that are reported that produce nanoparticles by they grow at optimal conditions. Therefore, the aim of the present study was to obtain cadmium sulfide nanoparticle synthesis by biomass of cadmium-tolerant fungi. Here we have 17 fungi isolates obtained from different locations from Hidalgo State, Mexico, and which 65%, 23%, and 12% came from different locations. For example, from wastewater polluted with heavy metals, wastewater sludge, farmland, soil irrigated with wastewater, and the isolates that we obtained were evaluated to their tolerance to different cadmium concentration. And as can be observed in table 1, the higher proportion of cadmium of isolates were able to grow at 100 ppm of cadmium salt, whereas only 2 isolates were able to grow above 500 ppm of cadmium salt. And also, as can be observed in red, are indicate 3 isolates that by microscopy were present morphological characteristic from Alternea genus, whereas 6 isolates corresponding to Aspergillus genus by microscopy with morphological characteristics. And the remaining isolates were discarded because they present this morphology. And here is important to note or to mention that the last year, Aspergillus genus was reported by cadmium sulfide nanoparticle synthesis ability. And on the other hand, Alternea was investigated by their ability to cut to silver nanoparticles synthesis. For cadmium sulfide nanoparticle synthesis and fluorescence spectroscopy analysis for the characterization of optical properties, we had to produce 3 different experimental. Here, the first are a biological control with fungal biomass and water. The second is a cadmium sulfide nanoparticle reaction where the nanoparticles were synthesized and contain fungal biomass, cadmium, sulfur, and finally inorganic control with cadmium and sulfur as reaction precursors. And by fluorescence spectroscopy analysis in a excitation wavelength of 365 nanometers, we found a band around 440 to 460 nanometers corresponding to amino acids and NADH enzymatic cofactor. And also we found a peak at 615 nanometers corresponding to antioxidants and metallothioneins involved in reduction process to synthesize cadmium sulfide nanoparticles. And finally, only in the cadmium sulfide nanoparticle reaction, we found the peak at 515 nanometers corresponding to nanoparticles. In all the isolates or in all the samples, we found the peak at 515 nanometers corresponding to cadmium sulfide nanoparticles. And here in blue are indicate the isolates that were selected to additionally analysis, and in red also are indicate the isolates corresponding to Alternea genus to additionally analysis, because only the 4 highest fluorescence responses were selected to another analysis. And yellow is indicated the peak at 515 nanometers. And finally, the dynamic light scattering or DLS and the transmission electron microscopy to size and morphology characterization of nanoparticles, we found in the samples particles with hydrodynamic radius around 200 and 1000 nanometers and corresponding to cell debris. And also we found particles less than 100 nanometers corresponding to conglomerate particles as can be observed in the micrographies at left side by TEM. And finally, we also found particles less than 10 nanometers corresponding to free cadmium sulfide nanoparticles as can be observed in right side of TEM by transmission electron microscopy. Here it's important to mention that the particles that we found are within the quantum dome regimen, and this regimen are characterized by a high stability of optical properties. And as a conclusion, we have that the cadmium sulfide nanoparticle synthesis by cadmium tolerant fungi was evidenced by fluorescence spectroscopy, dynamic light scattering, and transmission electron microscopy analysis. And it is what demonstrates that the obtention of particles smaller than 10 nanometers by these isolates are corresponding to aspergillogenous, whereas the isolate, the final isolate was correspond to Algenia genus. And for it is all for this work. This is the reference for the presentation. And thank you very much for your kind attention. If you have any question, I will be pleased to answer them. Thank you.
Thank you, Daniel. Okay, we are going to wait for a minute for the question of the public because the lag. But tell me, this is an interesting work of, I think, hours of laboratory. Can you explain a little bit how much time do you spend in your laboratory to obtain these results?
Okay, well, we spend around, if the work is quickly, we spend around 3 or 4 months, because the time of growth, the fungi are around 4 days, and also we have to realize the reaction synthesis that imply another day, and for example, to obtain one reaction of one fungi, we spend around 1 week. And but for example, if we have more experience, we could obtain around 3 reactions per week, but the time is a little
It is long.
long.
Yes, it is a long time. Well, we have a question of the public. Dagoberto Pulido Arias asked, what was the most challenging part of your research? Maybe the pollution.
Well, the can you repeat me the question, please?
The most challenging part of your research.
Okay. Well, the most important or the most difficult
yes,
was the maybe the characterization of particles, because the samples that we obtain are or they have a high concentration of cell debris, and clean these samples are very difficult, because in my microscopies or micrographies, the cell debris appear in the micrographies, and it's difficult to see the particles, or also is difficult found colloidal pH to found the particles free. And I think that this is the one of the most difficult things that we have been present.
Okay, many thanks. Many thanks for being here and for sending us your work for publication. Thank you. And well, goodbye. We'll see you in the closing ceremony.
Thank
And
you.
well, bye bye. Well, our next speaker, well, our next work is reducing power of curcuma longa extract and its influence on the synthesis of copper nanoparticles. The authors are Norma Gabriela Rojas Abelizapa, Itzel Alejandra Cruz Rodríguez, and Andrea Margarita Rivas Castillo.
Good evening.
Are you there? Hello, I think Alejandra, yes. Hello,
Yeah.
Alejandra.
Can
Well,
you hear me?
yes, we can hear you. We can see your presentation. And well, the stage is all yours.
Thank you very much. Oh, good morning, everyone. My name is Alejandra Cruz, and I am a student of Psicata Querétaro. And today I'm going to present the work that we have been doing at our lab, which was done in collaboration with both of my mentors, Dr. Norma Rojas and Dr. Rivas Castillo. The work is called Reducing Power of Curcuma longa extract and its influence on the synthesis of copper nanoparticles. Okay, well, so to begin with, I'm going to tell you about nanoparticles, which are very well known because of their size. And this can go from 1 to 800 nanometers. Here you can see an image of them. Actually, these are copper nanoparticles, but synthesized in a different way. And when we talk about nanoparticles, we can have different types of them. We can find polymerics, ceramics, and the most common or popular ones are the inorganic ones, which are metallic. And these ones are able to have unique electrical or magnetical and optical properties. And we can also find them in various sizes, structures, and geometries. According to the characteristics of our nanoparticles, we are going to be able to apply them in tons of things. But how do we synthesize the nanoparticles? Well, in order to do so, we can go through chemical synthesis or physical synthesis, which are the most common ones. But there is also an alternative that is called green synthesis. This is well known because of the use of bacteria, fungi, yeast, and also plant extracts. However, it is not well known how this works because of the metabolites that are involved during synthesis. Therefore, there are some discrepancies between authors. Ones say that the reducing power should be the one that is able to reduce the particles, while others say that maybe reducing power isn't all. So in order to prove this part, we base our work in the methodology that was early published by Hayarambu et al. In 2020. And they worked with curcuma longa extract, specifically with curcuma longa root, in order to create a green synthesis method for copper nanoparticles. So the aim of this work was to evaluate the influence of reducing power present in curcuma longa extract in the synthesis of copper nanoparticles. I'm going to tell you about the materials and methods that we implemented. First, we had to go through plant extract preparation. So for this part, we have to wash very well the roots. And once they were washed, we let them dry. And once they were dry, we got a fine powder. With this fine powder, we took 10 grams of it and we put it in contact with 800 milliliters of ethanol during 4 hours at 70 degrees. Once the process was done, we got around 50 milliliters of our plant extract, in this case, curcuma longa extract. Then first, green synthesis of copper nanoparticles. As I told you earlier, we took the methodology that was early published. And what we did was to change some things in order to see the influence of the reducing power. So in order to do so, we prepared a stock solution of copper acetate, and then we dilute the solution in order to get different copper concentrations on it. And we kept the curcuma longa extract the same in all of the synthesis. And after that, we had the synthesis, and we characterized through UVB's analysis, and we also performed a FRAP assay that was able to tell us a little bit more about the reducing power involvement. So here, and this would be the first part of our results, and these are about the synthesis of copper nanoparticles. And as you can see in here, in yellow, you will see copper acetate, which was our precursor material, and in gray, you will see the extract that we use. The blue band is the one that is showing us the presence of surface plasmon resonance, which shows us a peak specifically at around 447 nanometers. So we took this as a reference of a successful synthesis of copper nanoparticles for all of the rest of the studies. So once we had the reference and we knew that at 447 nanometers, we were going to be able to see the nanoparticle synthesized, we went through all of the different syntheses. And the first thing that we observed here was a change in color. As you can see, the lowest concentrations which were 100 and 200 milligrams per liter of copper, they have like really light colors, while concentrations above 300 milligrams per liter, they have a dark coloration. So according to the methodology that we followed, at this point, we only thought that copper nanoparticle synthesis was successful on concentrations above 300 milligrams per liter. But later, we performed the UVB's analysis in order to confirm if we had this surface plasmon resonance. And we saw that even though we had very light colors, we still were able to see the peak around 447 nanometers, which tells us that that syntheses are being performed here. Another thing that we observed was a difference in the amount of absorbance that we saw. For example, the lowest concentrations had the highest absorbances. And at this point, we also thought that it could be attributed to the amount of reducing power present in the synthesis. So in order to prove that, we performed the FRAP assay that I told you earlier. And the results, you can see it in here. As a reference, we took the curcuma longa extract, and then we analyzed all of the syntheses that we implemented here. And as you can see, the lowest concentrations, they still have a high amount of the reducing power, in this case, which is expressed as milligrams per liter of ascorbic acid. And when we go through concentrations of 300 milligrams per liter, it reduces more. And when we see the results for 400 and above, we can relate that no reducing power was detected through this assay. We correlate this data, and the result was minus 0.66, which indicates the following, that is in the conclusions. So first, that there is an inverse correlation between the reducing power, expressed in this case as milligrams per liter of ascorbic acid, and the concentration of copper acetate used during the synthesis process, which means that while the copper concentration used during synthesis increases, the reducing power decreases at the end of the synthesis. And finally, that reducing power of curcuma longa extract evaluated through a FRAP assay is responsible of copper nanoparticles formation from copper acetate. However, involvement of other metabolites are not discarded at this point. And that would be everything from me. So thank you very much. And if you want to ask something, go ahead.
Okay, many thanks, Alejandra. I'm sorry, I see Daniel. Well, we are going to wait a minute for questions for the public. But I have a question. Well, two questions.
Okay, go ahead.
One is, I see different colors, but the same concentration. It is a relation about the color and the concentration. Or what do you think is why the color is different, but the concentration is the same?
Okay, that's something that we were thinking that first, well, the first idea was that the light colors, I'm going to go there, we couldn't have a successful nanoparticle synthesis at that point because literature tell you that you have to get like dark solutions in order to successfully synthesize nanoparticles. But when we went through the UVBs, we realized that even though the color isn't dark, we still have the nanoparticle formation. So what we thought when we did the FRAP essay was that we realized that we still have reducing power present in here. So there is like an excess when we do not use all of the reducing power that we are expressing here as milligrams per liter of ascorbic acid. We can have these light colorations, but once everything was used is when we have like these hard or darker colors, and it is also related to the amount of precursor material, in this case copper acetate, that was used during synthesis.
Okay, very interesting.
And the other one,
yes. The other question. The other question is how much time all the process costs? How much time do you spend doing all this process from the dry to the synthesis?
Well, in this case, the drying process, I would say it was the longest period of time. It took around a week, more or less. And after that, synthesis was pretty quick. We got all of the synthesis like in 15 minutes. That's one of the advantages of green synthesis, that they can be done almost immediately.
And another question of the public, Jose Daniel Aguilar Loa asked, what was the easiest part of your experimentation and why?
The easiest?
The easiest and the difficult.
Okay, the easiest part was to put in contact my plant extract and the copper acetate. And I would say that the most difficult part was to analyze all the data that we got in order to understand what was happening in here, because there is very few information about how green synthesis work because of the complexity of it.
Well, again, many thanks. Well, thank you for your presentation, and bye-bye. We see you in the closing ceremony. Bye-bye. Okay, our next speaker is Bruno Flores Hernandez. Bueno, the authors are Bruno Flores Hernandez and Jose Santos Cruz. The title of the presentation is Development of a Method to Produce a Potential Transparent Conductive Material. The presentation is going to, well, the presenter sent us the video, so we're going to reproduce it. Go ahead.
Good morning, good evening, or good night. My name is Bruno Renato Flores Hernandez. I'm working with Professor Jose Santos Cruz. I'm gonna show you the results of the work titled Development of a Method to Produce a Potential Transparent Conductive Material. So, what I'm gonna talk about? Well, I'm gonna give a little introduction, then I'm gonna talk about the methodology, the results, and the conclusion. In this work, I'm gonna present a method for the preparation and characterization of graphene using 6 different commercial detergents, also sodium cholate using as to keep these materials, and the use of 2 different equipments, one for sonication and the other one a mixed reactor for shear exfoliation. Well, but first, what is the material that I'm gonna show? It's graphene. What is graphene? Well, it is an allotrope of carbon consisting of a single layer of atoms arranged in a two-dimensional honeycomb lattice. This material was discovered by Andrew Game and Novoselov in 2004. It has revolutionized the vision of materials, and also this is important because graphene has shown some outstanding properties like heat transfer, energy, and hardness properties. And this is the first two-dimensional materials that has opened a new world of nanotechnology. Nowadays, there are investigations, new investigation methods to help mass production of this material. But there is a problem because some of them are damaging the environment. So methods, for example, that involve peeling in aqueous mediums are those technologies are developed. So there is a huge potential opportunity for industrialization. In this work, it concerns the environmental properties, and well, then there are different methods to produce graphene. Some of them are peel exfoliation, the first method. There is also graphene oxide reduction, chemical vapor deposition, the use of laser, sonication, and shear exfoliation. The final two, those were the methods used for this work. First, sonication. What is sonication? Well, sonication is a process that provides an efficient use of energy in addition to the better synthesis of particles, and this is using mechanical waves. So it is also used in the jewelry, for cleaning jewelry, and there is also shear exfoliation. Shear exfoliation could be related to what a blender does. Sorry, it has the main possibility of it is a technique that has been developed, and it is also a good technique to improve the production of graphene because it has developed. Also, there is cheap comparing to other techniques in this work. Both shear exfoliation and sonication works with a exfoliation process. How does it work? Well, you have graphite and then you put it in a liquid environment with some solvent. Could be a polymer, an inorganic solvent, or you could use surfactants like those containing in soap, and with the action of the mechanical process, you can separate these graphite sheets and became into a graphene. And well, the methodology for this process is first the surfactant selection. What was the criteria for this surfactant selection? Well, according to Profeco, this is the Procuraduría Federal del Consumidor here in Mexico, there are a lot of detergents, but the detergents selected for this work were those which also were environmental friendly, because one of the things said is the impact of pollution for the process. Then there is also it has to be a relationship between the surfactant concentration to surface station with water. This material was used, and also we determine the variables of interest. So for the determination of variables, 6 detergents were used. Also three concentrations to related three concentration because the concentration is related to the surface tension of water. Then two devices were selected, one for shear exfoliation and the other one for sonication. For shear exfoliation, two levels of speed were selected, as shown in the slide. And for sonication, two levels of frequency were selected. What are the results for this work? Well, first in Raman spectroscopy. Okay, Raman spectroscopy is a technique used for the study of graphene because it can give us the footprint of this material, and this is related to the bands. I mean, the presence of the D band, the G band, and the 2D band are a footprint for graphene. And also each band gives different information. For example, in the case of D band, it is caused by a disorder in the structure of graphene. The presence of this disturbance of carbon show it in the band with high intensity. So we hope that there is a really good peak in this case. Also, the G band is related to stress on the sheet. So we want a straight G band. And finally, we have the 2D band, which is related to the behavior of frequency of the laser used for the analysis. So in this case, for sonication, the detergents with the best performance were 531 and 231. This is for sonication. The same behavior were shown in shear exfoliation, also with 531 and 231. Okay, so both detergents were with the best performance in both devices. Those samples were analyzed using transmission electron microscopy. The first, the figure 3, it shows that the 231 sample of shear exfoliation in the left side there are 4 images with different magnifications. A and B are graphene sheets with equal sonnet vat, but in the C and D there is also they show the performance of graphene. On the right side there is the diffraction pattern, and in the 1 to 3 below this diffraction pattern, it shows that if the 2 peaks inside are bigger or higher than the outside peaks, it is also a footprint of graphene for these samples. This behavior is shown in figure 3 and in figure 4 for the 5G1 sample. So in this work we showed that graphene can be achieved by those methods. So it has been demonstrated that graphite can be exfoliated to give few layers of graphene in this work with a concentration of 1 milligram per milliliter. But the work that has to be done in the next days, it's also the fabrication or the manufacturing of a thin film for this material. And with this material, we can use it, with this thin film, we can use it on optoelectronic devices. And this is the objective for this work. Thanks a lot.
Thank you.
Well, thank you. I'm sorry for this technical issue. It was all for our technical conference session of materials and manufacturing. I hope you enjoy it. And well, stay tuned for more conferences of keynote speakers. We have the workshops. Well, stay tuned. Thank you. See you the next. Bye bye.